Hey, how are you, class? Hope you're fine and well. So, today we are going to talk about cell physiology. First, at the beginning of this class, at the end of this class, you should be able to understand or define the term cell physiology, define what osmosis and division, and also state and explain the factors affecting osmosis and diffusion. So, cell physiology is the study of the functions of the cell structures. Let's talk about membrane structures and properties. A membrane is a surface structure which encloses the cell and organelles. Membranes regulate the flow of materials into and out of the cell or an organelle. Examples of membranes include the cell membrane, the tonoplast, that is the membrane surrounding the vacuole, the nuclear membrane, mitochondrial membrane, and the chloroplast membrane. So let's continue with the cell membrane. It has three layers, that is the two protein layers and a phospholipid layer sandwiched in between the two layers, that is the two protein layers and a phospholipid layer. There is a diagram. You should be able to draw it well and label it, because I set it in the exam, so you should understand it well, study in details. Let's continue the properties of the cell membrane. It is permeable. It has small pores allowing for the passage of molecules of small sizes into and out of the cell. The cell wall, however, allows small materials to pass through it. Hence, it is referred as to being permeable. Sensitivity to changes in temperature and pH. Extreme temperature and pH affect the cell membrane since it has some protein layers. Such changes alter the structure of the membrane, affecting its normal functioning. Possession of electric charges. It has both the negative and the positive charges, helping the cell to detect changes in the environment. These charges also affect the manner in which substances move in and out of the cell. Hope you are following and understanding what the lesson is all about. Let's continue the physiological processes. Please pay attention. The physiological processes, the ability of the cell to control the movement of substances in and out of the cell is achieved through the physiological processes such as diffusion, osmosis, and active transport. Let's start with diffusion. Diffusion is the process by which particles move from a region of high concentration to a region of low concentration. There we have we will be demonstrating a practical activity to demonstrate the diffusion process using a potassium permanganate. When a potassium permanganate is placed in a beaker and some water is added, you will see the particles moving, and all the purple color of the potassium permanganate 7 dominates the color in the beaker. The difference in concentration of particles within the region of high concentration and the region of low concentration is what we call diffusion gradient. Also, in this diffusion, we'll be demonstrating, like when someone in the classroom, let's say when Mary opens a bottle of perfume at the corner of the class, we will be able to smell that perfume, all of us in the class, because the particles have moved from the region of high concentration, that's from the corner, and it dominates the whole class. Now that's what we call diffusion. Let's talk about the role of diffusion in living organisms. Number 1 is absorption of materials. Mineral salts in the soil enter the root by diffusion, since their concentration in the soil is greater than the root hair cells. Digested food, that's glucose and amino acids, diffuse across the walls of the ileum into the blood for transport to the rest of the body. Gaseous exchange in plants and animals. In both plants and animals, respiratory gases such as oxygen and carbon dioxide are exchanged through simple diffusion, depending on their concentration gradient. So let's talk about gaseous exchange in plants and animals. That's the role of diffusion. Remember, it's our role number 2 in the case of diffusion. In both plants and animals, respiratory gases such as oxygen and carbon dioxide are exchanged through the simple diffusion, depending on their concentration gradient. Let's hope you are following. Pay attention. Factors affecting diffusion. Factor number 1 is diffusion gradient. Diffusion gradient, a greater diffusion gradient between the 2 points increase the rate of diffusion. Diffusion gradient, we have said a greater diffusion gradient between the 2 points increases the rate of diffusion. Surface area to volume ratio. The higher the surface area, the greater the rate of diffusion, and the lower the rate, the ratio, the lower the rate of diffusion. This means that small organisms expose large surface area to the surrounding compared to large organisms. Small organisms therefore depend on diffusion as a means of transport of food, respiratory gases, and waste products. There's a diagram there. You need to study it in detail, understand. It's until what we have talked about surface area. Please read it in details, understand it well, because sometimes it may appear in exams and you'll be asked to demonstrate what's all about. Thickness of the cell membrane and tissues. The thicker the membrane, the lower the rate of diffusion, because the distance covered by the diffusing molecules is greater. The thinner the membrane, the faster the rate of diffusion. We have said that the thicker membranes have lower rate of diffusion because the distance covered by the diffusing molecules. Thicker membranes have a high distance to be covered by the diffusing molecules. That's why the rate of diffusion in thicker membranes is low. Thinner membranes are faster because their membranes thin, and that distance covered by the molecules to diffuse, the distance is small. No, that's why they diffuse faster than thicker membranes. The size of the molecules. Small and light molecules diffuse faster than large and heavy molecules. Let's take, for example, a piece of chalk. One we crush it and the other one we don't crush it. We put it in a beaker and add some water. We can see that the particles that are crushed, they tend to diffuse faster than the large particles. Now that's why we say small molecules diffuse faster than large and heavy molecules. Temperature. Increases temperature increases the energy content in molecules, causing them to move faster. Let's take, for instance, we take the same piece of chalk crushed and put it in a beaker. We light up a Bunsen burner and place the one on a Bunsen burner and heat it, and the one don't heat it. We observe that the one on the Bunsen burner tends to diffuse faster. That's the piece of the color of that chalk will appear faster in the beaker that is put on a Bunsen burner than the one is left not heated. That's why we say that the rate of diffusion is faster when temperature is applied to a certain molecule, and the rate of diffusion is low when temperature is not applied to certain molecules. Osmosis. This is the process where solvent molecules, that's water, move from a region of low concentration to a region of high concentration across a semi-permeable membrane. The highly concentrated solution is known as hypertonic solution, and a lowly concentrated solution is called hypotonic solution. Solution of the same concentration are said to be isotonic. Osmosis is a special type of diffusion because it involves the movement of solvent water molecules from their region of high concentration to a region of low concentration across a semi-permeable membrane. Please pay attention to this type of concentration. I've said hypertonic solution, hypotonic solution, and isotonic solution. Please pay attention, begin and be able to differentiate and note the difference in each. There is a practical activity there. We'll be talking about it. Anyone will be having a problem with it, pass by my office and consult. Let's talk about osmotic pressure. This is the pressure which needs to be applied to a solution to prevent the inward flow of water across a semi-permeable membrane. This is the pressure needed to nullify osmosis. Osmotic pressure is measured using a sigmometer. Let's continue. We have said the osmotic pressure is measured using a sigmometer. Sorry, it's not indicated in our notes, but I will update it. Osmotic pressure potential. This is the measure of the pressure a solution will develop to withdraw water molecules from pure water when separated by a semi-permeable membrane. Let's continue with water relation in animals. Cell membrane of the animal cell is semi-permeable, just like in the whisking tubing. Cytoplasm contains dissolved sugars and salts in solution form. If an animal cell, let's say the red blood cells, is placed in distilled water, that's a hypotonic solution. Remember we talked about hypotonic solution, and we said an hypotonic solution is a low concentrated solution. So when a red blood cell of an animal is placed in a distilled water flows in by osmosis. The cell will swell up and eventually burst because the cell membrane is weak. The bursting of the red blood cell when placed in a hypotonic solution is called hemolysis. If a similar red blood cell is placed in a hypotonic solution, water is drawn out of the cell by osmosis. The cell will shrink, and the process is called crenation. Body fluids surrounding the cells must be therefore have the same concentration, so as to which found in the cell. There is also a diagram there. Read it and go through it keenly. I'll test it in the exam, so begin, pay attention. Let's talk about water relation in plants. When a plant cell is placed in an hypotonic solution, it gains water by osmosis and distends outwards. As the cell gains more water, its vacuole enlarges and exerts an outward pressure called turgor pressure. As more water is drawn in, the cell becomes firm and rigid, and it's said to be turgid. The cell wall in plant cell is rigid and prevents the cell from bursting, unlike in the case of animal cells. We saw that in the animal cell it bursts because it lacks the cell wall. Begin with that. I'll ask you a question, so pay attention and follow keenly. The cell wall develops a resistant pressure that pushes outward the inside, the pressure is equal and opposite target pressure, and this is called the wall pressure. There is also a diagram there. Go through it keenly and read it carefully. When a plant cell is placed in hypertonic solution, water molecules move out of the cell into the solution by osmosis. The cell shrinks and becomes flaccid. If the cell continues to lose more water, plasma membrane pulls away from the cell wall toward the center. The process through which plant cells lose water, shrink, and become flaccid is called plasmolysis. Let's talk about wilting. When plants lose water through evaporation and transpiration, cells lose turgidity, shrink, and the plant drops. This is called wilting. If water supply from the soil is inadequate, plants do not recover, hence permitting what we have just talked about is wilting. Role of osmosis in organisms. We have absorption of water from the soil. Root hair cells of plants absorb water from the soil by osmosis. F support. Cells of herbaceous plants which are less woody absorb water, become turgid, and hence they support the plant. Opening and closing of the stomata. During the day, gall cells synthesize glucose, drawing in water, became turgid, hence open the stomata. During the night, they lose turgidity, hence there is no photosynthesis. As a result, they shrink, thus closing the stomata. Feeding the insectivorous plants. These plants are able to change their target pressure on the leaves, which close, trapping insects which are digested to provide the plant with nitrogen. You know that insectivorous plants, these are plants which feed on insects. So you can note the role of, they feed through the role of osmosis. Then there is osmoregulation. In the kidney tubules, water is reabsorbed back to the body by osmosis. Factors affecting osmosis. You have concentration of evolutions and concentration gradient. The greater the concentration gradient between 2 points, the faster the rate of osmosis. Optimum temperature. Optimum temperature is the temperature that is minimized so as we don't destroy the membrane. So optimum temperature is required during osmosis. Not too high, not too low, but optimum. Active transport. This is the process that moves substances across cell membrane against a concentration. This process requires energy to move these substances across the cell membrane and involves substances such as amino acids, sugar, and main ions are taken in by living organisms through active transport. You have the role of active transport. Reabsorption of sugars and usable substances by the kidney. Absorption of some mineral salts by the plants. Absorption of digested food from the alimentary canal into the bloodstream. Accumulation of substances in the body to offset osmotic imbalance in a arid and saline environment. Excretion of waste products from the cell. Please pay attention to these roles of active transport. Read it in details, note it, understand it well. Pay attention to it keenly and be careful. Factors affecting active transport. Oxygen concentration, pH change in pH, glucose concentration, enzyme inhibitors. Hope you have been following our class. If you enjoyed, Please take notes of what you have learned today. Whatever you have noticed, understand it well, pass by my office. I'll be there always. Consult, ask for questions, clarification, and read keenly. The time left is for you to read, go through the notes, understand them well, and be careful in your reading. We'll be having our cut as usual, as you know, after each topic, having a small cut. Read, understand well, be careful in your reading, be careful in the diagram I said you read through them. You may see them in your cut. And in the case of notes, I ask the Marianne, pass by my office, take the notes as your free time, update them, and I'll check the notes in our next class. Note, we'll be having our cut in next class. Read carefully, revise thoroughly, and be careful in your reading. Diagrams must be well drawn, understand them, and be keen with those diagrams. Thank you. Bye bye. See you next class. Wish you well.